Hi, am I audible? You can just write it in the chat if you don't want to unmute yourself. Hello, am I audible? Welcome everyone. This is week 9 of the PMRF problem solving session for the course Basics of Crop Breeding and Plant Biotechnology. So let's go to the first question. The question is, which of the following is true? Option A is all eukaryotic genes lack intron. Option B is all eukaryotic genes contain introns. Option C is all eukaryotic genes contains UTR or untranslated regions. Option D is majority of eukaryotic genes contains intron. So there are four options given to you and you have to select which of the following is true. So eukaryotes, first I think you need to know what eukaryotes are, right? So the living world can be divided broadly into eukaryotes and prokaryotes, right? So what are eukaryotes? So just think about the answer if you know it or not. But eukaryotes and the other like classification is prokaryotes. So the living world can be broadly divided based on certain criteria into eukaryotes and prokaryotes, right? So organisms which have a nucleus, right, which is enclosed within a membrane, right, and other organelles are eukaryotes, right? So bacteria is part of the prokaryotic world, and all other organisms, uh, that is fungi, plants, animal kingdom, all these come under eukaryotes, where they have a true nucleus, that is a nucleus. The DNA is enclosed within a nucleus which has a membrane, right? A nuclear membrane, right? And they have other organelles too, right? Like the mitochondria, the endoplasmic reticulum, etc. So there is a difference between the genes that are present in eukaryotic cells and that is present in prokaryotic cells, right? So let's see what they are. So this is the one on the top that you can see is the eukaryotic gene structure, right? And the one on the bottom is the prokaryotic gene structure. So this is, let's first start with the eukaryotic gene structure. So in the 5 prime side, you have the promoter, right, with the enhancer or repressor. Okay, this is the regulatory gene, right, and this is the operon, right, where you have the promoter, you have gene, the operator, the different genes, terminator, etc. Right. So not all prokaryotic genes need to be in the operon form, but this example has shown you operon containing prokaryotic gene, right? So, and this is the eukaryotic gene structure. So you have distal control elements like the enhancer or silencer. You have the proximal control elements like the promoter. You have introns and exons, right? And you have terminator or enhancer or silencer, that is downstream regulatory sequences, right? So which of the following is true? There are four options given to you. All eukaryotic genes lack intron. All eukaryotic genes contain intron. All eukaryotic genes contain untranslated region, and majority of eukaryotic genes contain intron. So which of the following was true? Let's see what the correct answer is. So the answer is majority of eukaryotic genes contain introns. So like you can see, these are introns, right? And they are interrupting sequences that will be removed after processing of the RNA to form the mRNA, right? They don't uh, make parts that will go into the protein, right? So that is what intron means in this case, right? And why is all eukaryotic genes contain intron not true? Because there are certain genes like the ACT1 gene in yeast, right? So it's an example which has a gene which does not have any introns in it, right? So introns are not the name given to non-coding sequences that are found within genes in eukaryotic cells, right? So in prokaryotic cells, they don't have introns. Okay, so let's move on to the next question. The question is overexpression construct must contain which of the following? And there are five options given to you, and there can be multiple correct answers in this, right? So, option one is gene of interest, option two is operon, option three is positive selectable marker, option four is promoter, and option five is terminator, right? So, the question is overexpression construct must contain which of the following. So keep in mind that it means which is absolutely necessary in overexpression construct, right? So there are four options given to you, right? So let's take them one by one. So the gene of interest is the gene that you want to overexpress. An operon is uh, a unit of a genetic material, right? That are mostly found in prokaryotes, right? That contain, like we were seeing in the example before, that contain a cluster of genes, right? And this cluster of genes, it comes under the activity of a single promoter and operator gene, right? So, and these genes are mostly related to each other, or like they belong to maybe the same metabolic pathway, or they form the genes. Like these genes are responsible for, the production of a single kind of metabolite, right? So, that is why they are required. Their expression is required to be in a coordinated manner. That's why the operons exist, right? So, let's let's go back to the question. Positive selectable markers are selectable markers, like are when you transfer a vector into another cell, when you're doing DNA, recombinant DNA, right? So when you're transferring a vector into another cell, right? So you require positive selectable markers, right? And that is because you need to select the cells which have the plasmid or the vector in it, right? So promoter is like in this gene, the promoter here, right? Yeah, so that is where the RNA polymerase will bind, right? And the terminator is where the RNA polymerase will stop, right? So these are the five options given to you, right? Um, so um, let's see. Yeah. So the correct answer for overexpression constructs must contain which of the following? The correct answer is gene of interest, because obviously you need to have the gene that you're trying to overexpress, the promoter and the terminator, right? So that is because the mRNA or the RNA will be formed only if you have a promoter or terminator. You don't necessarily require a positive selectable marker, right? And you don't require a operon in this case. These are not compulsory. So the must containing things are gene of interest, promoter, and terminator, right? So let's just one second. Yeah, right. Let's move on to the next question. Question number three is introns containing genes should be cloned for bacterial expression purpose through which of the following process? There are four options given to you. PCR, using gene-specific primer and genomic DNA. Option B is PCR using random primer and genomic DNA. Option C is PCR using mRNA and gene specific primer, and option D is PCR using gene specific primer and cDNA. So, what this question tells us is that intron is present in the gene, right? So that means this is a eukaryotic gene because only those have introns, right? And this eukaryotic gene has to be cloned in a bacterial expression system, right? So bacterial expression systems don't have the mechanism to remove introns when they convert the um, genetic data into mRNA, right? So that's why you need to do some pre-processing to get this gene that can be cloned into bacterial expression system, right? So out of the four options, let's first see what the correct answer is, right? And then I'll explain it. So the correct answer is option D, that is PCR using gene specific primer and cDNA. So all the options had gene specific primers except option B, which said random primers. So you cannot use random primers here because for the specific gene to be amplified, right, you require primers that belong to that specific gene, right? So that's why you use gene specific primers, and instead of mRNA or genomic DNA, right? So mRNA cannot be used in PCR because it's RNA, right? And tag polymerase will only be functioning when you have a DNA template, right? So this is not the correct answer. Genomic DNA will have introns and exons, right? So that can also be used because if you use the genomic DNA to amplify the gene product, you will get mRNA with both introns and exons, right? So you don't want that because the introns have to be removed, right? So what you use is the gene specific primers and cDNA. So how that works is you have you use the mRNA of the gene that you want, right? But this mRNA cannot directly be used in PCR. So what you do is you use reverse transcriptase enzyme, and what it can do is it can make a, complementary DNA strand or a cDNA against the RNA, right? So this is the reverse transcriptase enzyme doing the function, and it is forming a DNA RNA hybrid, right? Okay. And after this, what happens is because this cDNA that is being formed, right, it can form a hairpin loop structure like you can see here, right? So when you use NaOH, right, and what happens is the RNA and the DNA will separate from each other, and the S1 nuclease can what it can wait, yeah, sorry. You use DNA polymerase, and since you have a complementary region of DNA double strand, right? Your DNA polymerase can act on it and extend this, right? So now you have a double-stranded DNA with a hairpin loop structure at the end, right? And then you can use S1 nuclease. So what that does is it removes this hairpin loop structure. So now you have a normal double-stranded cDNA molecule, and this you can use for cloning into bacterial expression system because you use the mRNA, right? And this mRNA doesn't have any introns, right? So this is a total completely processed RNA, that is the mRNA. So it has only the gene information, right? So you use PCR gene specific primers and a cDNA, right? So let's move on to question number 4. Question number 4 is you have to match the following, and on the left hand side you have four options: high auxin to low cytokine. Option 2 is low auxin to high cytokine. Option 3 is auxin and cytokine proportional to each other, and option 4 is 2,4-D, right? And on the right hand side you have three options: shoot formation, callus formation, and root formation. So you have to match the following, right? So that means what proportion or what plant hormone will be used for these three processes, that is shoot formation, callus formation, and root formation. So let's see the answer first. So the answer is D. That means one, that is high auxin and low cytokine, is used for root formation. Two is A, that is low auxin and high cytokine is used for shoot formation. Three is B, where auxin and cytokinin at the same concentrations forms callus, and 2,4-D also forms callus, right? So that is the correct answer. So whenever you have a plant growth media and you have high auxin and low cytokine in it, what happens is root formation. When you have low auxin and high cytokine, what happens is shoot formation. If you have both hormones in equal proportion, you have callus formation. And if you have high concentrations of 2,4-D, which is a auxin hormone, it is also forming callus, right? So the correct answer in this case is option D. So let's move on to question number 5. So question number 5 is, what do you mean by collagenesis? And there is four options given to you. Option A is formation of embryoid from callus. Option B is formation of shoot from callus. Option C is formation of root from callus. And option D is formation of both shoot and root from callus. So let's see what the correct answer is. The correct answer for this question is option B, formation of shoot from callus. So the term given for the process where you have shoot formation from callus, right? And like in the example before, that happens when you have low auxin and high cytokine in the media, and you place a callus in it. So that process is known as collagenesis. And if you form roots from callus, it is rhizogenesis, right? So let's move on to question number 6. Question number 6 is, which of the following is not true for exonucleases, right? And the name itself should give you an idea of what this is talking about, right? So there are four options given to you. Option A is RNA processing. Option B is causes cleavage from 5 prime end. Option C is causes cleavage from 3 prime end, and option D is proofreading. So which is not true for exonuclease, right? So the thing that you need to see here is the use of the word exo, right? And the opposite of this would be endo, right? So you have endonucleases as well, and these two perform different functions. So exonucleases usually act outside, from the outside of a DNA or RNA, right? And endonucleases act on the inside or in between DNA or RNA, right? So out of these four options, let's first see what the correct answer is, right? So, which of the following is not true for exonuclease is RNA processing. So, exonucleases do not perform RNA processing. They can, because it's not mentioned if it's a 5 prime exonuclease or a 3 prime exonuclease. We can assume that exonuclease is a broad term that is being used here, and they cause cleavage from the 5 prime end and the 3 prime end, and they are also used for proofreading. So DNA polymerase has 3 prime to 5 prime exonuclease activity, right? So what happens is, if when the DNA polymerase is adding bases to the template strand, it adds a wrong base, right? Or if you have, if it adds the right base, and some tautomeric shift makes it converts that base into a tautomer, and that causes mismatch pairing here, right? Like you can see here, what happens is the 3 prime to 5 prime exonuclease activity of the DNA polymerase will remove that mismatched base, right? And then again, DNA polymerase will add new nucleotides, right? So that is what happens. And RNA processing is when you have a eukaryotic pre-mRNA, right? And it has untranslated regions, it has introns and exons. So the process of converting this pre-mRNA to mRNA, right? That includes removing of introns, adding the 5 prime cap and the poly A tail, right? The 5 prime cap, the poly A tail, and removing of all the introns to convert the pre-mRNA to mRNA, right? That is called, that is the name for the RNA processing. I mean, that is what RNA processing is, right? So exonucleases do not perform the RNA processing, but they do perform the other three things, right? So let's move on to question number 7. Question number 7 is, which one of the following is true for RNAi gene silencing construct preparation? There are four options given to you. Option A is full length gene clone is needed, that is more than 1000 base pairs. Option B is part of the gene, that is 300 base pairs is needed. Option C is native gene promoter sequence is mostly needed. And option D is operon is needed. So what could the correct answer be in this case, right? Just answer. So if you have forgotten what the RNAi silencing is, so you either have a double-stranded DNA or you have a hairpin loop, double-stranded RNA or a hairpin loop RNA, and an enzyme called dicer will make small fragments of this, right? And then you have the risk complex formation, right? And the risk complex uses this small fragments of RNA, which it uses a single stranded part of that small fragment of RNA, and it will target a mRNA fragment which has a part of that sequence, right, that it has within itself, and cleave in that sequence. So in that way, it cuts the mRNA at multiple places wherever it can find the sequence. And since the mRNA has been degraded, that mRNA is no longer useful, and that gene has been silenced. So that gene, no matter how many times it produces that mRNA, most of it will be degraded by this risk complex, right? And that is used for silencing, right? So how this works is, in the risk complex you will have a single stranded portion of the RNA, and once this risk complex moves along mRNA, it will find the complementary sequence, right? And wherever it finds the complementary sequence in the mRNA, it will cut it, right? So that is how it works. And with respect to this question, the correct answer is part of the gene, that is 300 base pair is needed. So right here you can see that a double-stranded RNA is being broken into smaller pieces, right? So for for you to prepare a RNAi gene silencing construct, only a segment of the gene is required, that is mostly around 300 to 500 base pair long, okay, to and this will generate small interfering RNAs, right? So this portion is very crucial for the purpose, because that is what will act as a template for the production of the small molecules, right, that will target the mRNA to have the gene silencing effect, okay. Yeah, so operon is not needed. Native gene promoter sequence is also not mostly needed, and the full gene clone is also not needed, right? So the correct answer is part of the gene, which is around 300 to 500 base pairs is needed, right? So let's move on to question number 8. Question number 8 is, which of the following will not lead to direct haploid production by tissue culture? So diploid is 2n chromosomes, right? And haploid is n chromosomes, right? So which of these followings will not lead to haploid production by tissue culture, right? There are four options given to you. Option A is anther, option B is leaf cell, option C is pollen, and option D is microspore, right? So what could the correct answer be in this case? So let's see what the correct answer is first. So the correct answer is option B, leaf cell, right? So anther, pollen, and microspore, all three of them belong to the male sex organ, right? In plants, leaf is not part of any sex organs. It is 2n in nature, right? All the cells in leaves will be 2n in nature, and therefore it will not give rise to a haploid when you use it in tissue culture. But anther, pollen, and microspore, like, wait, you can see here, this is the anther, this is the pollen, and this is the microspore tetrad, right? So all these have are or have haploid cells in them, right? Or are capable, or in the case of anther, of producing haploid cells in certain conditions, right? So these can be used for haploid production in tissue culture, right? But leaf cell cannot, right? Yeah, in other words, leaves are somatic cells, right? They are not uh, uh, germinal cells, right? So that is why they cannot be used for haploid production, right? So let's move on to the next question. Question number 9 is, for the production of disease-free plantlets, which method is widely used? There are four options given to you. Option A is callus culture, option B is anther culture, option C is meristem culture, and option D is hairy root culture. Right? So you have to find from the four options which is used for disease-free plantlets. Right? So let's see what the correct answer is. So the correct answer is meristem culture, right? So the reason why meristem culture is used for disease-free plantlets, right? So assume that you have a plant which is a great variety, right? And you want to have more of these plants, but unfortunately, this plant has been infected by a certain virus, right? A plant virus. Now, if you use any part of the plant to produce new offspring, since it's a virus and they are within the cells, right? uh, you you can't use antiviral any you might not be able to use any antivirus um, chemicals to remove these viruses. So any plant that you have make from these cultures will also have the virus in it, right? So to avoid that, if you have such a plant, you use meristem culture to get a disease-free plantlet, right? So what this involves is uh, you aseptically culture the shoot apical meristem. So this is the shoot apical meristem. This is the leaf primordia. So that is the leaves that are being formed, and in between that you have the shoot apical meristem, right? Or you can also use other meristems. You can use lateral meristems, etc. to get, but mostly it is shoot apical meristem that is easily used, right? um, and you use this to get disease-free plants. So meristems are the very highly dividing or actively dividing parts of the plant, right? And so because they divide quite fast and they don't have, the xylem and phloem going to the meristems, and the viruses mostly use these transportation areas to get from one part of the plant to another, right? So since these don't have that also, so viruses do, and since they are actively dividing, they are less likely to have or harbor pathogens, right? And so this makes them a suitable source for having or obtaining healthy and disease-free plantlets. So everything else can have the virus, right? So that is why the others are not the correct answer, and the correct answer is meristem culture. So let's move on to the last question, which is question number 10. And the question is, a promoter deletion study was done in order to determine the binding site for a transcription factor on the promoter, which is activated on treatment with drug X. The following construct were made, and luciferase activity was determined and expressed in arbitrary percentages, right? So this is the gene, the luciferase gene, right? And depending on how much of this mRNA will be formed, right, you will have more of the luciferase activity and you will have more visible glow that can be measured, right? So if you remember from how luciferase works from the last module, right, and you have upstream of the gene, that is minus, so anything downstream of the gene, right, is in plus, anything upstream of the gene is in minus, right? So you have 1800 bases uh, upstream of the gene. You have 12,010 bases upstream the gene. In the second case, you have 868 upstream, and you have 432 upstream, like you can see in these four cases, right? So in each case, what is happening is you're removing a certain part of the DNA, right? So if this is what you started with, you remove certain genes to get to here, then you remove more genes to get to here, then you remove more genes to get to here, right? And you're trying to find out where the promoter region is, right? And you do that by deleting a little bit of the DNA and observing the activity to determine where exactly in, and you know that the promoter will be upstream of the gene, right? So it can be anywhere here, and you want to find out the specific location of where it is. So you do this promotion deletion study, right? And what you can see here is the controls are all the same, right? And if control is where you're not giving the treatment with drug X, right? So this uh, binding site for the transcription factor on the promoter is not activated because you're not giving it the treatment of drug X, right? So that means this activator is not working as such, right? So you, and the black bars that you see is when you have treatment with drug X. So you can see a very high activity for luciferase in the -1800. You have a slightly lower activity in -1210, and an even lower activity, but the same for -868 and -432, right? So the question is, sorry, the following assumptions were also made. Option A is region between -1800 and -1210 contain a binding site for an activator. Option B is region between -868 and -1210 contain a binding site for a repressor. Option C is region between -868 and -432 contain a binding site for a repressor. Option D is region between -1210 and -868 contain a binding site for an activator. Right? Wait. The hairy root culture is not part of this question. Sorry about that. Right. So let's see what the question is. So the question is, which of the following assumptions are correct? Option A is A and C, option B is A and D, option C is B and C, and option D is only D. So option A is A and C, option B is A and D, option C is B and C, option D is only D, right? So you have to choose which of them is correct in this case, right? So let's go back to the figure that you can see here, right? So the correct answer, I'll tell you directly, the correct answer is B, that is option A and D. So option A was stating that the region between 1800 and 1200, that is this region, okay, contains the binding site for activator. And option D, right? Yeah. Option D was that the region between 1210 and 868, right, also contained a binding site for activator, right? So how can you tell from these figures, right? So first you have the where nothing has been deleted part of, like the gene is there, and upstream you have -18 till -1800, right? So on treatment with drug X, right, you can see that the activity of luciferase is very very high, right? And once you delete the area between 1800 and 1210, you see that the activity goes down a little bit, right? So that means this region, that is between the 1800 and 21 the 1210, right? This area had an activator, and once you deleted that, you remove the binding site for that activator, and that is why the activity of luciferase decreased, right? Because the activation has also decreased because this site has been removed, right? Then option D was the region between 1200 and 868 also contained the activation site, right? So once you delete the area from 1210 to 868, you see an even more decrease in the amount of luciferase activity, right? So that means again, you had, if there was no more activation sites here, right? uh, like if the activation site was only between 1800 and 1200, what you would have seen is this part would also have the same kind of bar, right? But you don't see that. You see a decrease in this, right? And that is because 1210 and 868, between that region you had another activating activator site, right? Or a binding site for an activator. Once you go from, or you delete this portion, where you delete the portion between 868 and 430, you don't see any more decreases. So the amount of luciferase formed is the same, right? So that means uh, there is no more activator in that region, right? So from the study we can tell that this area and this area had activate binding sites for activators, right? So the correct answer is option B, which stated that A and C are correct. So this region and sorry, A and D, this region had a binding site for an activator, right? So that's all for today. I hope I was able to clarify any of your doubts, and any and all of your doubts. Thank you so much for listening.